platform which I've described. This is Jack.
I'm a Canada research chair in medical genomics. So basically what Farid talks about the menu, I make new recipes basically. I put new menu items on the card, so to speak, for you guys. And my interest is mostly driven by my interest. So it's a good way to do work. And more interesting, it's also it motivates you to try to bring some of the tests to today. Basically, we still do ELISAs and stuff that I don't think we should be able to do anyway. And now I also have a next generation sequencer in my lab where I can sequence all of you for, you know, if you have $50,000, I could do that for you too. But essentially, today I'm going to talk about Streptococcus Pneumoniae, which is a very important disease. And the reason I started doing that one is because Pfizer and well, Wyatt initially, I guess, had a beautiful vaccine called Prevnar that we give to all of our little kids, and that reduced mortality by about 25%. But the problem, every time, I'm in infectious disease, every time you introduce something new, it's like Jello. Huh? Infectious disease is like Jello in some ways. You press something and something else will pop out. And essentially, we don't see the first vaccine for Prevnar had 7 serotypes, and we don't see them very much anymore, but we see new ones. So now they're coming up with Prevnar 13, and I guess they'll go up and again and again. So essentially, we have to monitor the epidemiology of some of these diseases as those vaccines are introduced. And I've been doing this with my collaborator, Alain Gervais, in Switzerland as well. So why did we do this? Well, I think it's a medical reason. Somebody would pay me to do it as well, and it was a very big medical interest for me because I'm in a children's hospital. And essentially, the serotypes right now are done by an antibody assay, like we used to do an HLA assay and a quelling assay. It's cumbersome, extremely costly, takes a long time, and it's something I don't want to do. So essentially, we had to come up with something new. Those serotypes are defined by the sugar polymer that are on the cell surface, the capsule. And there's a lot of antigenic variability. If you think about HPV, you got 91 serotypes. You got about the same for Streptococcus pneumoniae. And to identify which one, you have to look at multiple targets because they're so similar that you have to check at many spots to figure out what you got. And the way we do these assays, and we mimicked it on what we used to do for a respiratory viruses assay, we make a multiplex PCR. Sometimes we have 60, 70 primers and probes in the same tube and amplify the whole mix, and then try to automate the last part using the Infinity instrument. So we basically wanted to do this automation. And the process is simple. We use a multiplex. I'm afraid to press some of those buttons. Is there a pointer in there or not really? I'll do my finger. So basically we do a multiplex PCR, and then when that's done, we basically load it on the machine. You have the primer extension that adds your fluorophore, and then there's the washers, and then the primer, and then when the fluorophore is at the right spot, you know what you got at that specific spot, and then you read it. It's a little bit more complicated to read when it's a multiplex assays, especially for these, because you have to devise some sort of automate system, expert system, to be able to actually read what you got. So the gene we targeted, you have to think about that quite carefully. Every Streptococcus pneumoniae will have this gene, pneumolizin. So basically that comes your positive test. So at least when you do the test, you will know you got a Streptococcus pneumoniae. If you had autolizin, which is also a very common gene, I think you get the specificity from 96 to about 98%. So you're at least you're sure of that. And then when you add all of these other genes, then I can start telling you what serotype, what genotype you actually have for Streptococcus pneumoniae. And here's how it went. Initially, we wanted to be able to monitor the 7 of them that were in the Prevnar 7, basically the vaccine that was given about nearly 10 years ago now. It's on the market. The new version Prevnar 13 is coming. It's been approved in the U.S., at least in Canada for sure, this year. And we added these ones. So now, for 5 years, we'll have to monitor all of these serotypes precisely to see if there's escape mutant, new things that come into it, and if there's niche replacement, so one serotype coming in to replace another one. And as we devised these things, obviously we wanted to try to make the best we could, so we can actually detect 51 genotypes, serotypes, specifically as individual serotypes. And then all of the others we can detect at least some of them as groups. So all together we cover practically, well, all of them. So, and we started testing it, and we had some quite interesting results. Those are the serotypes we tested. Most of the time we actually can identify a precise serotype, close to 80%, or we can assign as a group, 100%. And as I told you, we can always be quite sure about the specificity when we use the general genes that are typical to Streptococcus pneumoniae, to autolysin and pneumolysin. So that works very, very well in our hands right now. So basically, this is how we interpret some of the results. It's a graphical. These are all the serotypes, and then these are how they're related to each other. And if you have one probe that's positive, this one, they will tell you which one it is. And that's how the expert system decides who you are, basically, as a Streptococcus pneumoniae. And we did some samples. We got them from children. We tested we have a lot more, but when we prepared that slide, we're at that stage. We have tested straight from blood, some from CSF. These were children from Cameroon, because we don't even know what serotype circulates in these countries where Streptococcus pneumoniae is actually a big problem, and also nasopharyngeal samples. And we detected 26 different serotypes quite readily, and our concordance with sequencing, so still the gold standard, at least in my lab, is about 96%. So for this particular part, well, we think we have a nice assay right now to do a multiplex molecular diagnostics for Streptococcus pneumoniae. We can detect 51 serotypes uniquely right now, and the others as groups. The assay is pretty robust because we build in some extra. We have enough spots on the microarray to build some redundancy. If one probe fails, another one can give you the information that you require. So that's something that Ferrit didn't address, but you can build fail-safe systems in there by putting extra probes and be on the safe side to know what you've been doing. We've actually tested that with real clinical samples that were flown in from Cameroon and straight to our lab, so it works quite well. And I think it'll be very useful in a clinical setting, obviously, especially after it's mostly a public health issue right now, but it could become also important diagnostics because certain serotypes are associated with greater resistance. So I think it'll be very important to monitor this precisely. The other assay we've been working is an evolution of our initial assay, which was a respiratory virus panel assay, which was built with the same intent of covering everything. But this one, I guess marketing pressure somewhat wanted to drive, let's just test flu, but do it really thoroughly, quickly, and all of it very precisely. And basically we design an assay where we could detect all type A influenza, type B influenza, and then again build in redundancy to be quite sure that everything's worked well. The pandemic H1N1. We can detect H1, H3, H5 quite individually very well. And also we added in the N fragment, we added a resistance to Tamiflu, basically the only drug that we use right now. And everything was controlled with armored RNA internal control, so we know that the extraction worked well, everything worked well, and we have a good assay, a robust assay. So the process, I can go through a little bit, but I guess these people can do it way better than me and actually show you exactly. But we always start from nasopharyngeal aspirates or throat swabs or the like. So as long as you can get good quality RNA or DNA, you will be able to perform these assays. and I guess our next speaker will address that quite readily. Reverse transcription, multiplex PCR, and the process that's automated on the instrument. The assay, we've been doing this for a while, and we can build assays in about 6 to 8 months doing a decent clinical trial and have something that we can use at least at research purpose. And then if you push it, we can get quite a decent clinical assay. Here you're looking at ratios over background. So I think this is the background. So you can see that fivefold here. We detect all of these different strains of influenza A quite well, some of them very, very well, and all of the strains from the influenza B. And you can also see that your internal control is good all along the way. So you can really control your work, and if you have some ambiguity, and everything that's done in our lab, we sequence anyway. But it's performing very well for this particular assay. And when we look at all of our strains, and some of them had resistance in them, we detect them at about 500 PFU per mil. So that's pretty good. I mean, the titers for these are very high, and it's most of the time, if you get a patient that has some symptoms, we detect it. If they present too late, then it goes down, and no assay in the world will detect anything anyway. Or by that time, it's too late to do anything. Same. This was for H1N1. It was performed very well. Actually, we tried to increase the sensitivity for obvious reasons. Those that are not supposed to be detected were not. Those were detected had a PFU of 100 PFU per mil, so that's excellent. And obviously, we didn't see any cross-reactivity with strains that should not be detected. Sorry. And the other one that circulates also is the H3N2. And that one also, maybe a teeny bit more work on this one, but around 500 PFU for this one as well. So this assay can be multiplex many patients onto the same. So it's a very high throughput. And we demonstrated here that you can actually put a drug resistance on these. So it can affect diagnostics, because with H1N1 within a month, at least at our hospital in Canada, we had patients that had H1N1 that were Tamiflu resistant, so why give Tamiflu? So these assays are very easy to do when they were quite well packaged in some ways, and it's easy to run. Very high throughput. We validated everything with reference strain. All the probes at 500 PFU per mils and about 100 PFU for H1N1. And the next steps is to do some clinical studies. We're waiting for the next flu season to do this. And the one good thing is that at this stage, this particular assay is being transferred to AutoGenomics for pushing it through. Thank you.
I'm Bo Fessenden. I'm CEO and President of Natural Molecular Testing.